Hello, my name's Jan Knodel. I'm the Extension Entomologist for North Dakota State University. This presentation is on honeybee pesticide poisoning and toxicity. Honeybee pollination is valued at an astonishing $14.6 billion in the U.S. In fact, pollination by honeybees is vital to the production of many crops as water and sunlight. 1/3 of our daily diet relies on honeybee pollination. Apples, almonds, sweet cherries, plums, and prunes are examples of crops that require cross-pollination between varieties in order to produce a crop. Bee pollination is necessary for the production of cucumbers, squash, pumpkins, and melons as well. In some field crops, self-pollinated field crops, there is a growing body of research that shows the presence of honeybees and native pollinators actually increased yield and quality. Honeybees are also valued for their honey production, and North Dakota just happens to lead the U.S. in honey production. It is valued at $70 million in 2010. We're just starting to understand why honeybees are so sensitive to pesticides, and this has to do with the identification and sequencing of the honeybee genome. We now know that the honeybee genome is deficient in the number of genes encoding detoxification enzymes. These are the enzymes that break down pesticides. So in other words, honeybees are much more susceptible to pesticides than other insects. This has negatively impacted the beekeeping industry for as long as pesticides have been used. Despite our dependency on honeybees for pollination of crops in the U.S., colony numbers have continued to decline 45% over the past 60 years. Most of the honeybee losses from 1966 to 1979 were attributed to organochlorine, organophosphate, carbamate, and pyrethroid pesticide exposures. Efforts to restrict pesticide applications during bloom have provided some relief. However, residual activities from some pesticides have never been effectively addressed. Colony losses were especially severe from 1981 to 2005, with a drop from 4.2 million to 2.4 million colonies. This has been attributed to the introduction of parasitic honeybee mites. The tracheal honeybee mite was introduced in 1984 and the varroa mite in 1987. In the picture there, you can see the red spot, which is the varroa mite. Another reason is the introduction of two new classes of systemic pesticides, the neonicotinoids and the phenylpyrazoles. These have replaced many of the older pesticides used for insect control. We also have the change in the development and use of genetically engineered crops. A study in 2008 analyzed 25 independent studies, concluded that Bt proteins used in genetically engineered crops to control lepidopteran and coleopteran pests do not negatively impact the survival of larval or adult honeybees. In contrary, it has actually benefited beekeeping by reducing the frequency of pesticide applications on crops protected by Bt, especially corn and cotton. On the other hand, the switch to genetically engineered crops with herbicide resistance has eliminated many of the blooming weed plants in the field along field borders and irrigation ditches. This has been implicated in negatively impacting honeybee health by poor nutrition. At a recent meeting I was at last fall, the North Dakota Honeybee Keeping Association, I asked the beekeepers how much they felt pesticide poisoning was a problem in their honeybee business in North Dakota, and the majority, 85%, said it was. Then I asked them if they had filed a formal complaint with the North Dakota Department of Agriculture about pesticides killing honeybees in the last 5 years. Fortunately, 68% said they had not, although 15% thought about it and 18% said they had. Then I asked why pesticide poisoning was a problem in bees, and they said generally they felt the main reason was a lack of understanding about pesticide poisoning in bees. The second reason is a lack of communication between producers and beekeepers when pesticide applications are necessary. And the third reason was a lack of understanding about the importance of the bee industry. Then I asked them which crops are the most concerned for bee pesticide poisoning, and sunflower was at the top with 43%. And this is probably because there's a large group of economically important insect pests that attack sunflower during flowering, such as banded sunflower moth and red sunflower seed weevil and sunflower moth. The second top crop was alfalfa, which pollination is very important during flowering for alfalfa seed production. The third crop was canola, and the concern there was the use of neonicotinoid seed treatments in the canola. Most of the canola, over 90%, is treated with insecticide seed treatment belonging to the neonicotinoid class. Insecticide toxicity. In the U.S., risk assessment related to agrochemical use and registration follows specific guidelines mandated by the Federal Insecticide, Fungicide, and Rodenticide Act. Despite the importance of honeybees, effect of the pesticide exposure on colony health has not been systemically monitored, and the Environmental Protection Agency does not require data on sublethal effects for pesticide registration. The standard method for assessing pesticide risk was to determine the LD50 of the insect pest, and then the second step was to determine the effect of the pesticide on beneficial arthropods such as honeybees and run an LD50 test. The LD50 is defined as the exposure level that causes 50% of the population exposed to die. And as mentioned previously, in honeybees that only included acute adult toxicity or mortality. The toxicity thresholds are highly toxic, acute LD50 of less than 2 micrograms per bee, moderately toxic, acute LD50 2 to 10.99 micrograms per bee, slightly toxic, acute LD50 of 11 to 100 micrograms per bee, and non-toxic, acute LD50 of more than 100 micrograms per bee. Highly toxic pesticides are defined as materials that kill bees on contact during application and for 1 or more days after treatment. Bees should be moved from the area if highly toxic materials are used on plants the bees are visiting. Here's some examples. Going to spend a little bit of time talking about methylparathion pencap M. This is by far the most potentially damaging pesticide for honeybees because it's packaged in microencapsulated tiny capsules of methylparathion. These capsules are about the size of pollen grains, which contain the active ingredient, methylparathion. When bees are out in the field, these capsules become attached electrostatically to the pollen collecting hairs of the insects, and at time, because of their pollen-like size, are picked up by the honeybees. Because of the microencapsulation, there's also a long residual. This slow release feature allows the methylparathion to be a potential killer for several months, and it can go back into the hive. At the present time, there's no way to detect whether bees are indeed poisoned by microencapsulated methylparathion, so beekeeper could potentially lose his whole hive if it is poisoned by this insecticide. So therefore, we strongly recommend that this pesticide not be used when there's a possibility of honeybee exposure. Now I'd like to talk about encevalerate or Asana XL. As you see, Asana is highly toxic to bees when exposed directly during an application. However, once the foliar residue has dried, the hazard is insignificant due to a bee repellent effect. This bee repellent effect reduces bee visitations for the first day, and then it will return to normal. By following these guidelines, producers can see effective insect pest control with little or no interruption of bee activity to pollination-dependent crops. Research has confirmed that bees do not carry Asana XL back to the hive in quantities that either weaken the hive or predispose it to losses during the winter. In addition, dried spray residuals of Asana XL will not become resuspended or dissolved in dew, unlike some other pesticides, further reducing the hazards of Asana XL to bees. I'll also point out thiamethoxam, which is in the neonicotinoid class. This is a very popular insecticide seed treatment called Cruiser, and it's found on many of our field crops: soybean, sunflower, dry beans, potatoes, sugar beets, and others. Moderately toxic pesticides. These are materials that can be used with limited danger to bees if not applied over the bees in the field or the hives. This depends, though, on still using correct dosage, timing, and method of application. Here are some examples. Spend a little bit of time talking about Carbaryl or 7. 7 is one of the United States' most widely used insecticides for a variety of insect pests. It's also one of the most toxic to honeybees, depending on the formulation used. So the bees can be poisoned with Carbaryl, and it usually takes them 2 or 3 days to die, and they appear inactive. It allows them time to take that contaminated nectar and pollen back to the colonies. And again, the XLR formulation is fairly safe because it's moderately toxic, but other formulations like a dust formulation, if applied during bloom, can cause large numbers of bee mortality, and it has been responsible for disastrous kills. So again, check the label. The formulation is very important and can affect the toxicity to bees. Relatively non-toxic pesticides. These are materials that can be used with few precautions and a minimum of injury to bees. And fungicides are included in this group, like captan, copper, and sulfur. There's also other modes of actions, like botanical insecticides. Azadirachtin is dissolved or derived from a tree grown in India, and called neem. And pyrethrum is derived from the chrysanthemum flower. We also have insect growth regulators like dimlin, which is popular for the control of grasshoppers in field crops. The dimlin disrupts the young grasshopper from molting from one stage to the next, so it's not applied at the adult grasshopper, but only the immature stage. Confirm at the bottom is also another molting disruptor, which prevents the production of ecdysone, which is the molting hormone. And this is registered for control of Bertha armyworm in canola, for example. We also have Bacillus thuringiensis, or BT, which is a naturally occurring soil bacterium that controls insect pests. Again, we need to check the pesticide label, check the environmental hazard section of the label for any bee pesticide restrictions, and look for words like actively visiting or foraging in the field, or visiting and flying through the field. Just a little bit more on the neonicotinoid and the phenylpyrrozole insecticides. These insecticides are extensively used in the U.S. on field, vegetable, turf, and ornamental crops. They can be applied as seed treatment, soil treatments, or foliar sprays applied directly to the plant foliage or the flowers. They're systemic in the plant, so they are translocated throughout the plant and are translocated to the pollen and nectar during blooming. The use of these newer systemic insecticides has become prevalent in the U.S. As systemic, these pesticides are present in all plant tissues, including the nectar, pollen, and other plant exudates. Honeybee exposures to these compounds is very different from that of traditional pesticides, where acute toxicity was the primary concern. Instead, honeybees at all stages of development may be chronically exposed to sublethal doses of these compounds. The consequences of these new modes of exposures have not been extensively considered in regard to pesticide regulation in the U.S., although the EPA is currently reviewing the status of these compounds. While the translocation of neonicotinoids into the pollen and nectar of treated plants has been demonstrated, the potential effects that the level of neonicotinoids found in pollen and nectars can have on bees remains unclear. There has been some study looking at pesticide exposure and interactions with plant pathogens to harm honeybee health, and they found that honeybees that were treated with imidacloprid, a neonicotinoid, and fed noseum spores suffered reduced longevity and reduced glucose oxidase activity. We need new methods to assess honeybee toxicity, especially as it pertains to some of the newer chemistries. Acute toxicity tests on adult honeybees may be particularly ill-suited for the testing of systemic pesticides because of the frequency of exposure bees are likely to experience in the field. Chronic feeding tests with using whole colonies may provide a better way to quantify the effects of systemic insecticides. Some other tests may be using honeybee larvae in vitro tests, which could give us some idea on any learning disruption or behavioral effects. Certainly, pesticides is one of the players in colony collapse disorder, although no one pesticide has been clearly associated with causing colony collapse disorder. There is evidence that additive and synergistic effects of multiple pesticides exposures are harming bees. However, there's other players involved, such as viruses that have been identified, Also the parasitic mites that we talked about, and then the miticide sprays that they use to control the mite parasites. They're applied directly to the hive, as you can see on the photograph on the right. And a new cause has recently been identified, and that is a phorid fly that parasitizes bees. Parasitized bees by the phorid fly become disoriented and turn into zombies, and they fly out into the night to die the next day. So there's multiple factors causing colony collapse, which pesticides is just one of the factors. We'll now watch a short video to see techniques that you can use to help mitigate pesticide bee mortality. This video is about bee safety and protecting honeybees from pesticide poisoning. Honeybees and native pollinators are a vital part of agriculture food production and should be protected from pesticide poisoning. The value of bee pollination is estimated at $14.6 billion in the United States. With a reduction in the number of domestic and wild bee colonies due to colony collapse disorder and other diseases, the value of honeybees and native bees for pollination has increased. This increases the importance of protecting bees from pesticide poisoning. Bees are attracted to blooming field crops such as sunflowers and even weeds such as dandelions in the field for nectar and or pollen. Because bees forage for up to 2.5 miles or more from the hive, all beekeepers within 2 to 3 miles of the area to be treated with insecticide should be notified several days before the insecticide is to be applied. In North Dakota, the names of beekeepers can be obtained by going to the North Dakota Department of Agriculture's website. Bee pesticide hazards can be reduced by following these guidelines. Know and communicate with beekeepers about hive locations. Use economic threshold and other integrated pest management strategies. Economic thresholds ensure that pesticides are used only when crop losses prevented by pesticide use are greater than the cost of the pesticide and the application. Use pesticides with low toxicity and low residual to bees. For example, avoid using dust or wettable powder insecticide formulations because they generally are more toxic to bees. Evening or early morning applications are the least harmful to bees because fewer bees are foraging. Do not spray when winds can cause pesticide drift. Use all pesticides in a manner consistent with the label directions. The environmental hazards section of the label may include specific restrictions that protect bees. Words that describe pesticide restrictions are while actively visiting or foraging in a field, and visiting or flying through a field. Typically, bees are actively foraging when there's daylight and temperatures are above 60 degrees Fahrenheit. Thank you for learning about how to protect pollinators from pesticide poisoning.